Ladies and gentlemen, first of all, thank you for your kind introduction, and I'd like to thank the organizers for giving me the opportunity to talk about the common mucosal immune system. On the first slide, I have an outline of roughly what I will be talking about, specifically functional advantages of secretory or milk secretory IgA, origin of IgA, induction of immune responses, common mucosal immune system, and mainly the future studies, which I think should be performed to exploit the mucosal immune system of the mammary gland for the prevention of certain diseases. First slide deals with the mucosal, with advantages of secretory IgA. Not too many people realize that IgA in milk is in the form of dimeric or tetrameric molecules. In other words, 2 or 4 monomers linked together through disulfide bridges, which enormously enhances its neutralizing activity. There are a number of papers published, mainly from Great Britain and from our laboratory, clearly showing that, if you have monomer, you have 2 antigen binding sites. And if you have dimer, you would say 4, and therefore double the protective effect. Actually, it's not that way. The bonus effect of multivalency indicates that actually in dimers, you have 100 times or even 1,000 times greater neutralizing activity than you would have in a monomer. So this bonus effect of multivalency is enormously important, particularly for IgA in external secretions. Generally speaking, IgA is quite resistant to proteolysis, which is absolutely necessary for the survival, as we heard from Dr. Ogra, through the intestinal tract, which is full of different enzymes. Mike Russell showed beautifully, he was lucky that he had patients with multiple myeloma with known antibody activity, IgG and IgA double myelomas, and showed very beautifully that IgA is a strongly anti-inflammatory isotype of antibodies, which is particularly important in mucosal secretions or mucosal tissues. As Per Bransek published in Nature previously, IgG in the mucosal tissue can activate, after joining the antigen, can activate the complement cascade, influx of polymorphonucleoleucocytes, and damage of the mucosal tissue, which is not the case if IgA is present, because IgA outcompetes IgG complement-mediated activation. The other interesting, and that's a relatively recent development, showing that IgA molecule has actually 2 important functions. One, with a specific antibody activity as a specific classical antibody, and second, as a carrier of innate immunity due to its carbohydrate moiety. Secretory IgA is very heavily glycosylated. Some 10-12% of total molecular mass is contributed by carbohydrates. And as you will see on the next slide, these carbohydrates can very actively interfere, at least in vitro, with the binding of bacteria or even viruses, microorganisms, to the receptors on epithelial cells. So it's a wonderfully important function. A broad spectrum of antigen activities which reflect the exposure to the external environment, inhibition of bacterial adherence as well as viruses to receptors on epithelial cells, and through the binding of factors of innate immunity, such as mucin, lactoferrin, and lactoperoxidase, it enhances or focuses the activity of innate factors on a given antigen, which is recognized by antibody, which is associated or linked with these substances. This is a molecule of IgA. Here we have FAB fragment, FAB, this is dimer, so 4 antigen binding sites, adaptive immunity, recognition of specific antibodies. Here is the FC region, which contains, besides J chain, also so-called secretory component, which is a polymeric immunoglobulin receptor expressed on epithelial cells. And in yellow, orange, and red, you have lots of carbohydrates which are attached to all components except for the light chains. And these carbohydrates, which sort of stick out, can very effectively bind to bacteria through sialic or microorganisms through sialic acid, mannose, and so on, and inhibit the interaction with epithelial cells. This is just a slide. We heard about it, but not in differences in species, IgG in milk of pigs, cows, horses, and so on, FcRn receptor transported through epithelial cells, and for the first 36 hours of the life, picked up by epithelial cells in the gut of these animals, and the IgG molecules enter the circulation. So the classical IgG in milk of these species. In humans, there is very little of IgG, as Ray Ogra was showing in previous slides. Dominant is IgA in a dimeric or tetrameric form. It has to be in a polymeric form and containing J chain. Without it, it would not be picked up by the receptor on epithelial cells. And it stays in the gut lumen or on mucosal surfaces. There was one paper many years ago by Ray Ogra showing that actually a little bit of, or trace amounts of IgA from milk are absorbed into baby circulation, but it was a very small amount and certainly not significant with respect to the protection. So this is a very marked species difference as it is. Molecular properties of IgA and why we worry about where it comes from. As I said, in milk, dimeric or tetrameric form, 60% roughly, dimeric 40%. Tetrameric in serum, 95 to 99% is monomeric IgA, two heavy 2L chains. In humans, we have two subclasses of IgA, IgA1 and IgA2, and only chimpanzees and gorillas, and to a certain degree orangutans, have two IgA subclasses. All other species, mice, rats, tigers, elephants, you name it, have only one IgA isotype, which structurally resembles human IgA2, not IgA1. IgA1 is a newcomer for a number of reasons. When you look at the distribution of, by the way, funny thing, in lagomorphs, rabbits, North American pika, and so on, there are 13 or 14 IgA subclasses, 1,3 or 1,4, and only one IgG. So that's an unusual situation in these species. In milk, we have roughly 50% of IgA1, 50% of IgA2. In serum, 85%, 15%. All of these data clearly indicate or suggest that the IgA in milk is not coming from the circulation. IgA is produced locally, as you will see, by plasma cells present in a lactating mammary gland, while IgA for the circulation is from the bone marrow. This is an interesting story. Many years ago, some smart dentists from the United States, from New York, irradiated peripheral blood of patients with acute lymphocytic leukemia, put it in the drum with radioactive cesium, so the arterial blood went in and it returned into the vein. And then they measured the levels of immunoglobulins in saliva and in the circulation. Circulatory IgG, IgA, IgM, no change whatsoever, because these cells did not migrate through the circulation, while IgA in saliva disappeared, clearly indicating that through the irradiation, they killed the cells which were on their way to populate salivary glands where they would differentiate into IgA-producing cells. The other interesting point concerns the probability of minimal contribution from the circulation. Many years ago, Dr. Delacroix from Warman's and formerly Heremans's laboratory in Belgium injected radioactively labeled IgA, monomeric, polymeric, IgG, albumin, and so on, into the patients, into volunteers, and collected external secretions, particularly gut secretions. And to their great surprise, only very small amount, 1% of total IgA in external secretions was contributed from the circulation. We did an identical study using anti-idiotypic antibodies. Dr. Hiromi Kubagawa from Max Cooper's lab raised anti-idiotypic antibodies to myeloma proteins. These were patients with IgA or IgG myeloma or IgA macroglobulinemia. And he produced antibodies which recognized specifically idiotypic determinants of these monoclonal antibodies. And we collected saliva and examined for the presence of these myeloma proteins in it. And again, 1% of total IgA in saliva was contained, although it was polymeric IgA with J chain. It was not effectively transported. Why do I even mention that? Well, using modern technology, one could easily produce now in large quantities polymeric IgA with given antibody specificity and think, well, if I inject it intravenously, I'll protect, because it's polymeric IgA, transported, I'll protect external secretions, including milk, by the way. And obviously, for these reasons which I mentioned, it's not going to happen, because even polymeric IgA from the circulation does not contribute to the pool of IgA in saliva. So that method of enhancing, passively enhancing immunity in external secretions, probably including milk, would not work. This is from a paper by Dr. Brancek, where he evaluated density of antibody-producing cells in different tissues, including the mammary gland. As you can see on the slide, this is a number of cells per square millimeter. So you can see that mammary gland, lactating mammary gland, he had only 2 lactating mammary glands. We'll return to that problem later. Roughly the same as in a parotid gland or submandibular gland. Surprisingly, lacrimal glands were full of IgA and IgD-producing cells. And indeed, after serum, tears have more IgA than any other external secretions. And of course, colonic mucosa, full of IgA-producing cells, very high density of these cells. I apologize for this slide. It didn't get transferred properly, but I will say it at least about IgA and IgA1 and IgA2 antibodies. When you evaluate levels of total immunoglobulins in milk, you will see tremendous variability, depending on the day of collection, how you collect it, and how you process it. Usually, as I said, in our hands, roughly 50% IgA1, IgA2. In the hands of British investigators, 60% of IgA2, 40% of IgA1. Why is that important? Well, just like with IgG subclasses, IgA1 and IgA2 also exhibits specific antibody activity dependent on the type of the antigen. So IgA antibodies to the protein antigens, bovine lactalbumin, beta-lactoglobulin, you name it. Anti-protein antibodies in IgA isotype are usually IgA1, while antibodies to antigens which contain polysaccharides, and we looked at meningococcal, gonococcal, and pneumococcal polysaccharides, and LPS. Those antibodies are more present in the IgA2 subclass. I think that's relatively important, particularly for the intestinal tract and sensitivity to proteolysis, because IgA2 is more resistant to proteolysis than IgA1. IgA1 can be cleaved by a family of enzymes called IgA1 proteases produced by a number of pathogenic bacteria. Now, induction of antibodies in milk, local and systemic immunization. As you can imagine, particularly in milk industry, lots of attempts have been made to induce specific antibodies in milk of cows, sheep, and so on. Well, they were immunized either through the duct of the mammary gland or injection into the proper tissue of the mammary gland, and induced some antibodies, but interestingly, and look at the species too, interestingly, most of these antibodies were of the IgG isotype. The best paper that I could find was published actually here in Switzerland many years ago in the Journal of Immunology by Dr. Licha, who showed very nicely in rabbits who have which have IgA isotype as a dominant antibody in the milk, that when she immunized locally in the mammary gland with or without adjuvant, the dominant response was IgG, not IgA, and there was a significant local inflammation. So local inflammation into the secretory glands, including mammary gland or salivary glands, which has been done in a number of studies for immunization against dental caries and so on, is not a particularly useful method because of the induction of local inflammation and low IgA responses. Immunization by the systemic route. I called Ray Auger actually to ask him if he knows of any papers where people who were immunized systemically had good antibodies in milk. And he referred to 2 of his papers with his collaborators in Journal of Infectious Diseases, where a rubella vaccine given systemically, subcutaneously, induced some antibodies in milk of few lactating ladies they had. Relatively late after the immunization, it was not an immediate response. But I did not find, for example, a study where lactating women would be immunized systemically with the influenza virus, and somebody would look at antibodies to the virus. An interesting paper which I found, which I found in the literature concerned the differences of immune response in milk in different groups of people living under different conditions. Swedish women were immunized systemically with Vibrio cholerae vaccine, and they did not generate antibodies in milk. They had good antibodies in the circulation, IgG and so on, but not in milk. But when the same vaccine was given to women in Pakistan, in other words, in an environment where Vibrio is relatively frequently present, and these women usually have been exposed to Vibrio. When they immunized systemically, they did get some antibodies of the IgA isotype in milk secretions. So if you are previously mucosally exposed to a given antigen and then immunize systemically, you may induce some specific IgA antibodies in external secretions. I know about Vibrio, whether some other antigen would behave the same way, we do not know. This is a summary of studies performed for a number of years, particularly initially in the laboratory of our great friend and colleague who unfortunately died a few years ago, Dr. John Sebra, who demonstrated in his classical paper with Susan Craig, published in Journal of Experimental Medicine, that cells which form antibodies in mucosal tissues originate from so-called inductive sites, in this case, in Peyer's patches in the intestine. Peyer's patches, of course, have been in the literature for a number of reasons. Originally, some people thought that they were equivalent of bursa of Fabricius of birds and considered it as source of B cells and so on. And indeed, in some species, such as sheep, rabbits, the birth of B cells is not in the bone marrow like in humans, but it is in these mucosal sites, sacculus rotundus, and space-like structures where B cells are born. So these are primary organs where B cells are generated, which is not the case in humans. But Dr. Sebra, when they transferred cells from rabbits, they homed or selectively populated back the gut, the lamina propria, or other mucosal tissue. So this was one of the most important papers published in mucosal immunology in those years. These cells expressed, as was shown a little bit later, and I'll talk about it, receptors which direct or conduct where the cell will go into what tissue based on the expression of so-called homing receptors on surfaces of lymphocytes. And they are recognized by the ligands on endothelial cells of postcapillary venous, emigrate from the circulation and populate these tissues, where they terminally differentiate under the influence of T cell products or epithelial cell products into IgA-producing cells. And many, many studies since that time have been done. I would remind us of one really very important paper by Paul Montgomery, who clearly showed that in rabbits who were immunized orally with dinitrophenylated pneumococcus, mammary gland or milk from these orally immunized rabbits produced secretory IgA specific for the hapten, that is for dinitrophenyl. So he was the first one to show that indeed, oral, 1973, that the oral immunization of rabbits with this hapten on a carrier induces secretory antibodies in milk. So what evidence do we have of a common mucosal immune system in humans, that is system where inductive sites are source of cells which subsequently populate remote effector sites? What evidence do we have? As I will show you, and Ray showed it a little bit earlier, it's a spectrum of antibodies in milk, which reflects the exposure of the recipient to oral bacteria, food antigens, and sperm. This is an interesting story. Please, I'm not a sexologist, but we worked on anti-sperm antibodies. And to our great surprise, human milk from healthy women or even those who were infertile had relatively high, 40% of women, had relatively high titers of antibodies to sperm, and it was secretory IgA. What was the stimulus? Obviously not a local immunization, obviously not a systemic immunization. Intravaginal, which reproductive immunologists say that's the induction where antibodies are induced. Well, a number of studies since that time have been performed showing that actually genital tract, female genital tract, is extremely poor inductive site. And local stimulation in genital tract doesn't help. But then if you look at the sexological literature, some 80 to 90% of heterosexual couples engage in oral intercourse and some 40% in occasional intrarectal intercourse. And in both situations, of course, sperm is deposited in the sites of inductive sites, because in the rectum, as you will see, there is so-called rectal tonsil, which resembles the accumulation of Peyer's patches and therefore is the source, and I will demonstrate that, of cells which populate other mucosal tissues. So apparently sexual practices greatly influence and are the reason for the presence of antibodies against unusual antigens, sperm, in high titers in human milk. So one has to be read a little bit more than just how these antigens enter the human body. Detection, another reason, or another evidence for the presence of these antibodies, determination of antibodies including in tears, genital tract secretions after oral or intranasal immunization, and I'll show you a number of these studies. And then detection of antibody-secreting cells in peripheral blood. You can catch these cells which will populate mammary glands or salivary glands because they express unique set of receptors on their surfaces which direct their way. I think this slide I can skip because Ray showed us actually even more antigens than I have on this one. And the only one he didn't show was sperm, but I already talked about it. So we'll look at the next slide, which is common mucosal immune system in humans. Orally administered alive attenuated poliovirus vaccine work in New England Journal of Medicine by our chairman here, Dr. Ray Ogra, and his collaborators, Dr. Carzon, and I don't know who else was on paper. What they used, poliovirus vaccine given systemically, Salk vaccine, and Sabin vaccine given orally. And then they collected, unfortunately, none of the immunized persons were lactating females, but they collected nasal secretions and duodenal secretions. And Ray, in his paper, was the first one to demonstrate that indeed systemic, mucosal immunization will induce antibodies in external secretions of the nose and of the gut, while the systemic immunization will not. And these antibodies were IgA. So that was one of the landmark papers in the history of immunization and differences between mucosal versus systemic immunization. We followed with this work in our lab by orally administering Streptococcus mutans. This is a bacterium which causes dental caries, but we had a strain against which we did not have pre-existing antibodies. The antigen was put in the gelatin capsules, enterically coated. We ate it and then collected tears, saliva, intestinal fluid, and in one case, one of the volunteers was a lactating mother, so we had also some milk, and demonstrated very nicely that by ingestion of the antigen, we can generate in parallel antibodies in all of these external secretions, although there was no local stimulation. So our tears had plenty of antibodies against the oral bacterium, and obviously that was due to the fact that the cells were disseminated. And later on, Dr. Cecil Cherkinsky, who was in the lab and published a paper in PNAS, he characterized these cells, the appearance, when they appear, 6, 7, 8 days after the immunization, how they disappear, when the antibodies appear, and so on. So he provided not only humoral, but also cellular parameters of these immune responses. In the next study, which we have done in collaboration with Dr. Kantele, we orally or rectally and I explained why rectally, because of the rectal tonsil as an inductive site. With Salmonella typhi T121A Swiss vaccine here, produced in this country, against typhoid fever. And we immunized orally or rectally and induced antibodies in tears, saliva, and so on, as I will show you in one of the next slides. So again, oral immunization or rectal immunization generated generalized immune responses in secretions of the glands which were removed from the site of immunization. And since that time, a number of studies have been done with intranasal, sublingual, and nasal administration of vaccines, particularly influenza virus vaccine, to induce specific antibodies in external secretions. So here we have a scheme of the common mucosal immune system. Inductive sites, these are the principal ones, nasal or respiratory tract associated lymphoid tissues, mainly the work of our former colleague, Dr. Bienenstock, and gut-associated lymphoid tissues present dominantly in a small intestine, but as I mentioned, also in the rectum as a rectal tonsil, as the inductive site. Cells from these sites migrate into the circulation through thoracic duct, and you can catch them there, actually detect the antibody formation and so on, and then they populate various effector sites, such as respiratory tract, gastrointestinal tract, and so on. There are some differences, I think it's the next slide, yes. Some differences with respect to the source of or origin in the inductive site and the homing to different effector sites. As you can see from the nasal-associated lymphoid tissue, dominant receptor is alpha-4 beta-1 chain, which is a dimer on surfaces of lymphocytes, which populates these tissues. Please notice, and interesting, this is a slide from Dr. Per-Bransek. Please notice that there is no mammary gland included, because nobody has ever looked at the homing receptors in human, which are expressed on these cells which populate lactating mammary gland. From the gut, the dominant receptor is alpha-4 beta-7, which directs these cells to go to small intestine, large intestine, and so on, and partly to the genital tract. So, effective sites of immunization. What does it mean for vaccinology? As I mentioned, local responses, there are marked differences between the sites as well as species, but generally speaking, it's not very effective. Applying antigens on site where there is no inductive tissue present, that is certain types of epithelial cells, B cells, T cells, and so on, does not give a good responses. Systemic, as I mentioned earlier, it depends on the type of immune response and previous experience of the recipient of the vaccine. If previously exposed mucosally, you may generate some immune responses. And then the combination of mucosal and systemic route. Common mucosal immune system. Well, we have now several vaccines in humans. There are 6 vaccines currently available given by the mucosal route. Live polio, rotavirus, influenza virus, which is given intranasally in children. It was discontinued for 2 years, but I think last year it was again reinstated. The vaccine is effective in children, not particularly effective in adults, because we do have pre-existing antibodies, which may interfere with the effectiveness of the vaccines, that is infection with the live virus and generating the immune response. Adenovirus for children and Salmonella typhi bacteria and Vibrio cholerae vaccine originally developed in Sweden by Cecil Czerkinsky and what is it? Name, it will come. There are a number of veterinary vaccines available for Jan Holmgren, sorry. Number of vaccines for different species of animals, chickens, whatever. An interesting point is oral vaccines in fish industry, which is a tremendous problem. Because as you will know, salmons and a number of other fish are kept, are not caught in the ocean, but they are kept in farms. And these animals are immunized by the oral route, by a given pathogen and mixed to the feed, and they immunize them and generate antibodies not only in the circulation but also on surfaces of the animals, of the fish. There are many advantages to mucosal vaccines. As you see on this slide, it's inexpensive. You don't care if there is LPS contamination. You don't need syringes, needles, you don't need doctors to inject it, or nurses. Mass immunization within 1 day orally or whatever way, intranasally, you can immunize hundreds of thousands of people. Disadvantages of mucosal immunization, very low absorption of antigens. We do not have a very good adjuvant usable in humans, which would enhance the immune response. And pre-existing immunity greatly restricts reuse with the same antigen to the effective way. On the next slide, I think, which is one before the last, common mucosal immune system, human mammary gland. We don't have a whole lot of data on mammary gland with respect to the various routes of immunization and cells which are there, because to obtain samples of the gut through biopsy, that's not a large problem. But to obtain tissue from a lactating woman is very difficult. But as I mentioned, we have indirect evidence, presence of antibodies to everything you can imagine. Well, it won't go back. Nope. Well, I think I'm pressing the... No, that's the next one. Oh. Anyway, there was a paper which I wanted to mention, published by Dr. Ogra's colleagues, Randy Goldblum and Nene Hansen, who was a co-author on it in Nature. What they immunized lactating women with a special strain of E. coli and collected milk and collected and separated cells and fluids. And they induced very nicely antibodies to this strain of E. coli. Yeah, I did, but that didn't work. Oh, here. Golden fingers. So E. coli, they immunized them and induced specific antibodies in milk. And more surprisingly, when they took cell fraction from that milk and did Yernes plaque assay, you may remember what it is, erythrocytes coated with antigen, mixed whatever in gel, then you add complement and develop number of plaques. Up to 10% of total cells produce specific antibodies. Well, that was a great challenge for us because we worked on milk and on milk cells, and we wanted to know what kind of cells were responsible for the production of such a tremendous number, unusual number of cells producing antibodies. And on top of it, they could develop the plaques not only with anti-IgA, but also with so-called anti-secretory component, which is a receptor remaining associated during the transepithelial transport. So we spent lots of time on characterizing these cells, and with the help of a number of people, we showed that actually in human milk, we do not have antibody-secreting cells. B cells are present, and as Dr. Ogra showed, in relatively small numbers. The main cell in the milk is macrophage polymorphonucleoleucocyte, and only roughly 10% of total cells are lymphocytes, and most of those are T cells, not B cells. But B cells are certainly present, but they are not secreting antibodies. We examined these cells histologically, right Ginza staining, immunofluorescence with anti-IgA, anti-IgG, and so on. And to our great surprise, by immunofluorescence, the cells shine not only for IgA, but also for secretory component and for both subclasses. Plasma cell can produce only one, IgA1 or IgA2, but not both. And Itaro Moro, who spent 3 years in the lab, looked at hundreds of cells from milk by immunoelectron microscopy, and he couldn't detect a single plasma cell there. And then we analyzed the cells from the plaques, from the Yernes assay, by looking at the morphology of the central cell. And again, there were no plasma cells there. And most importantly, the plaques could not be inhibited, their formation, by addition of cyclohexamide, because cyclohexamide stops biosynthesis of proteins. So if you add, before you plate it, if you add to the cell cyclohexamide, you prevent the formation of plaques, which was not the case with milk cells. But anyway, we analyzed these B cells for their phenotypes and their responses. If you add IL-6 to B cells, they differentiate very promptly within a day or two into antibody-secreting cells. Colostral B cells didn't. Poquid mitogen, which is a T-cell dependent mitogen which induces in peripheral blood in humans, not in mice, but in humans, generation of antibody-secreting cells, plasma cells, and so on. Well, you have to have T cells there, and there are T cells in milk, but nothing happened. So we added T cells of the same donor from the peripheral blood. The cells did not differentiate. And finally, you can transform T cells with Epstein-Barr virus, and you can generate cells which secrete IgG, IgM, IgA. When we did that with milk, generally it was not successful. The cells were not transformable, and in those few individuals where we did get transformation, the dominant isotype was IgG, IgM, and very little of IgA. So obviously, these cells were not present there. Yes, antibodies were correct, but the cells which were carriers of IgA is Itaru Moro showed by electron microscopy, were polymorphonuclear leukocytes and macrophages. This is a staining for IgA, and you see IgA is in very nice granules within phagocytic cells. This is milk globule, again IgA in the middle of that globule, and this is a classical plasma cell where you have beautiful endoplasmic reticulum, polyribosomes secreting immunoglobulins. So the IgA, which is present in cells, is associated with phagocytic cells which do express receptors, Fc alpha receptors, on their surfaces. Dr. Pitard and others speculated that this IgA associated with cells is important in release and protection when the cell dies in the intestine. So we calculated the amount of total IgA, free IgA, and IgA associated with the cells. Cell lysate and measured by ELISA, or at the time radioimmunoassay, and found out that actually only 0.004% of total IgA is associated with cells. Yes, they may have IgA, but the amount in comparison to the free IgA, which we have normally in milk, is really a negligible amount of IgA. Last two slides. This is a study which we have done with Dr. Kantele and some other studies from the literature, looking at the induction of antibodies in milk after various routes of immunization: nasal, sublingual, oral, rectal tonsil, or intravaginal. And as you can see, all of these routes, to a different degree and depending on the antigen, induce antibodies in various secretions. Vaginal route is ineffective, except for cholera toxin B subunit. Antigens given intravaginally stimulate minimal immune responses, but we have absolutely no idea how, for example, intranasal, sublingual immunization would induce antibodies in milk, or rectal immunization antibodies in milk. So this is something which still has to be done. In the study with Dr. Kantele, we used a commercially available Salmonella typhi T121A vaccine given to women orally or rectally. They were not lactating, but we could collect tears, saliva, nasal secretion, all of these fluids, and serum. And as you can see, there were differences according to the same antigen, but differences in the immune response in different compartments if the vaccine was given orally or rectally. Rectal vaccine, to our great surprise, stimulated excellent antibodies in tears and in nasal secretions, and of course in rectal fluid, as you would expect. Oral immunization, very nicely in saliva and in vaginal fluids. So this is the last slide which I have, which sort of summarizes what we don't have and perhaps should address in our future studies. Which mucosal immunization routes are effective in the induction of antibodies in milk? We don't know that, because except for oral immunization, intranasal, sublingual has not been evaluated. What are the phenotypes and responses of B cells in mammary gland? Again, this would direct us to the route of immunization, because we know alpha-4 beta-1, alpha-4 beta-7. So it would determine which route of immunization may be more effective, whether intranasal or oral or rectal. We don't know that. Duration of immune responses after different immunization routes in milk, that is, we don't know that either, except for few antigens we looked at. And racial differences. This is a point which we are addressing relatively recently because of a disease called IgA nephropathy. It's the most common glomerulonephritis in the world, except it's absent in Africa and among African Americans. And nobody knew why. And so we looked through the literature, I will not go into IgA nephropathy, but to say that simply we do not have data in the literature dealing with the maturation immune responses depending on the racial association. We do not know whether IgA system or mucosal immunity in African Americans developed the same way as in Caucasians. We do not know, I couldn't find a single paper dealing with the levels of IgA as related to the maturation or age of the children. In white children, we know basically no IgA adult levels achieved in puberty. 1 or 2 years old kids don't have much IgA anywhere, maybe in secretions, but not in the circulation. But we really don't know anything. We don't know anything about milk of white versus African Americans or African black women, which may be important. And of course, one interesting point will be to determine the immunization of mothers when the antibodies in milk start to appear and how long they will persist. So I'll end right there. Thank you for your attention. We are doing okay.
Wonderful. Thank you, Jerry. That was a superb talk. Are there any questions? We have plenty of time. Let me ask you for... Oh, go ahead.
So, where is the...
Can you use this microphone?
Yeah. So, where is the secretory IgA synthesized that gets into the mammary gland?
It's produced locally by plasma cells distributed in the mammary gland, which are adjacent to the epithelial cells. The product, polymeric J chain containing IgA, binds to the receptor on epithelial cell. The receptor remains associated with transcytose in 45 minutes roughly, and released on the other end as secretory IgA. In other words, secretory IgA is product of two cells. Of the plasma cell producing IgA and J chain, and epithelial cell which expresses this so-called sacrificial receptor, polymeric Ig receptor, which remains associated with IgA. It gets cleaved proteolytically just before the secretion and gets into the milk. So product of two cell types. Is that the answer? So as I said, polymeric IgA from the circulation you would expect would also be picked up by polymeric Ig receptor and would go into milk or saliva, but it doesn't. We don't know why. Per Brandsek speculates, and I think correctly, that because of the locally produced IgA, it sort of outcompetes the IgA from the circulation, which is in minimal quantities in healthy individuals, so that there has no chance to get into the external secretions, into milk or saliva.
So there's two cells. If there are two cells, Jerry, where are they coming from?
Oh, well, epithelial cell is...
No, but what is their... Are they coming from the inductive site in the gut, mucosa? Or
no. Epithelial cell is a natural cell which is there to begin with. But the plasma cell, as I try to... they come... precursors of plasma cells come from different inductive sites. And the big question is, what is the dominant inductive site for milk, for mammary gland? My personal feeling, although I have no data, would be that it's a gut. Because after all, the product of the mammary gland ends up in the gut of the kid. And therefore, the milk must reflect the antigenic experience of the mother and therefore protect the child against the same antigens. Yes?
Go ahead. Right here.
I'm Dr. Sarwar Firdaus from Bangladesh. You have talked about the mucosal vaccines, and we have oral polio, rota, Vibrio cholerae, and salmonella typhi oral vaccine available in our country. But you have mentioned two other. One is influenza, and another is adenovirus mucosal vaccine. Yeah.
I mentioned those.
Do you have these two vaccines in Europe, or do you use this vaccine, mucosal vaccine in Europe? We do not have influenza and adenovirus oral vaccine or mucosal vaccine in our country. Do you have
influenza vaccine given intranasally, which is alive, attenuated, cold-adapted, reassortant virus developed originally at NIH by Dr. Brian Murphy and others, is available in this country.
Is it available in Europe?
I don't know whether it's available in Europe. I'm saying it's available in this country again because some 2 years ago, because of reduced effectiveness of the vaccine, it was discontinued. It was used like for 5 years. Then somebody published a big paper saying that it didn't show a whole lot of protection, and the vaccine was discontinued. But it was reinstated roughly 2 years ago. You are a pediatrician. I don't know whether you reinstated again, and vaccine is available, and it's recommended for children as an intranasal live attenuated vaccine. So it is available. Rotavirus vaccine, there are 2 of them available in the United States, given by the oral route. Adenovirus vaccine is given only to the military personnel. It's not generally available to everybody, but certain types, as you well know, adenovirus is a tremendously heterogeneous, many strains of the virus, but some of them are more common, and there is oral vaccine, attenuated adenovirus, given to the military personnel. So those vaccines are available. And Salmonella, which is a Swiss vaccine, is also available in the United States.
The oral vaccine is available. The rotavirus vaccine is available worldwide. Alan?
Jerry, a major problem with young infants is that the mothers who are lactating are given antibiotics because of strep.
Mm-hmm.
And that disrupts the initial colonization process, which leads to all kinds of problems. Why can't we immunize mothers so that they have antibodies against strep in the milk, and then not have to use the probiotic?
That was the last point of my presentation. That we simply need to know if we can immunize the mother before she starts lactation, and before she starts feeding the kid. What is the kinetics and what kind of antibodies can be induced in milk to protect the kids?
Haven't they actually shown that you can do that, or not?
Not that I know of.
Okay.
I haven't found a paper which would clearly show that if you immunize mother with whatever vaccine, I don't know which vaccine it would be since there are only 6 of them. So I don't know which one it would be.
Lactating mothers were immunized with rubella vaccine. Actually,
They were immunized what?
with rubella vaccine.
No, but that's systemic. That's mucosal.
But they do seem to transfer some degree of immunity in breastfeeding babies.
I mentioned that. I cited your paper, yes.
Any other questions? Yes.
Thank you. Just a comment regarding the source of B cells. Going to the mammary gland in animal model in the mouse one, they show that if you inhibit CCL28 that is expressed on epithelial cells in the large intestine, you block migration of
I'm very well aware of this paper, primarily from Dr. Michael Lamb's laboratory and many others, showing that indeed in mice, the cells are generated in the gut and migrate to the mammary gland. So I'm aware of those papers, many of them, Weiss Carrington and Michael Lamb and God knows who else. So in animals, that's okay. In rabbits too, but in humans, we just don't have the data.
No.
We assume that it's that way too.
The same,
Do you have any data on progenitor or stem cells, immunological
no, but
activity?
Dr. Tlaskalova, who's right after me, will tell us about that. I don't have any data.
You don't have any
No.
human data?
None of those.
Any
Since
other
we have a minute, let me tell you a story
have 10
about
minutes.
Nene Hansen, because I knew him very well too. He was a great friend and great colleague. And he was working on IgA and went to New York, to the Rockefeller University, in the laboratory of Dr. Henry Kunkel. And I went there several years after him. And the discoverer of IgA, Dr. Joseph Heremans from Brussels, came to Rockefeller University to show to Dr. Kunkel how to isolate IgA, zinc sulfate precipitation, gel filtration. So they grabbed Nene Hansen, bled him, and Joe Heremans purified IgA. Nothing.
He had no idea.
No, wait. So they grabbed another guy from the lab, John Rocky, big, Nene Hansen was tiny, small guy. John Rocky, huge, small. Bled him, Heremans, isolated IgA, isolated nothing. And Henry Kunkel said, well, guys, you are both IgA deficient, which they were. Now realize the probability of catching 2 people with IgA deficiency in a row, 1 out of 5 to 700 Americans are IgA deficient. So, and 2 in the row. So Nene Hansen was diagnosed by Henry Kunkel that he was IgA deficient. So there is another, I think, funny story about him, about Nene.
Well, the interesting thing is both Joe Harrimans, Tom Tomasi, Lars Hansen, and Jerry Mustiki worked at Rockefeller with Henry Kunkel
Yes.
at different times.
A great experience, yeah. Well, thank you.
Anyone else had a question? Yes.
My question is, my name is Dr. Syed Mazbabak from Bangladesh. My question is, oral polio vaccine is being rapidly replaced by IPV, by newer vaccine.
Yes.
So, oral polio vaccine is still available in Bangladesh, in third world countries, in mass vaccination.
Yes.
But do you suggest this oral polio vaccine in future for use? Because mucosal vaccine is told by Professor Mitchell, so I want to know that.
Yes, I think the best answer of all people on polio vaccines will be given by Dr. Ogra. Certainly, polio vaccines, or live oral polio vaccine has many advantages, particularly in developing countries where you immunize few kids and you immunize the whole village, because it gets into the stool, into the water supply, and so on, and you immunize the whole crowd. In the United States or Europe, in some countries, such as Denmark, they inject the vaccine, then they give polio. I don't know whether they still do it, but they used to do that. In the United States, now basically all the polio vaccination is with killed, that is Salk vaccine. Ray, you say it better than I.
Well, no, I think he's right. But the story of polio vaccine is a little bit more complex today than it was 50 years ago or 40 years ago, because we had a lot of polio with all three serotypes. But today we don't see type 2 poliovirus in the wild. So you have type 2 has been essentially eliminated all over the place. And presence of type 2 has often been associated with interference with type 1 and type 3 vaccines. So that has created problems in the use of oral OPV vaccines in many parts of the world. Now the tendency has been to use bivalent polio vaccine orally, which is very good, and then you can boost them with subcutaneous, with Salk vaccine, with very good immunity in most mucosal surfaces.
Yes, thank you.
Anyone else? No? Thank you.